when we reduce the irrigation, normally the mycorrhizal colonization increases. Okay? In soil, we also found differences in irrigation. Okay, so the irrigated treatments had more soil propagules than non-irrigated ones, but in this case, the inoculation treatment was not significant. So the inoculated and control plants were similar, statistically similar, in when we analyzed soil. Okay, so as a preliminary results and conclusions so far, remember that we are not finishing all the experiments yet, but we found that no-tilling or the regenerative agriculture treatment increases the cauliflower production in the regenerative crop. The total soil fungi tended to increase after the cauliflower crop, which is an interesting result. The molecular quantification of arbuscular mycorrhizal fungi in roots is highly correlated with the morphological count, so we use the molecular count because it's easier for us to measure. The water regime influences production in cauliflower and lettuce, but not in tomato, in the mycoplot. The arbuscular mycorrhizal fungal inoculation had no effect in the production of the mycoplot. The arbuscular mycorrhizal fungal root and soil colonization in lettuce increased under reduced irrigation in the mycoplot. And arbuscular mycorrhizal fungal inoculation enhanced root colonization in lettuce, even in the presence of native fungal symbionts. You have to remember that arbuscular mycorrhizal fungi are naturally present in the soil, but in this case, the extra inoculation also increased the colonization of the plants, even when the native arbuscular mycorrhizal fungi are there in the plantation area. And finally, let me introduce all the members of the AgriRegenCat team in a meeting here in Torre Marimón, and the participants of the Sustainable Crop Protection Program in the Centre de Cabrils, Carla Biel, she's here with me, Nuria Real, Elisenda Casteller, Montse Prat, Mari Carmen Bellido, and myself. Okay, thank you very much for your attention.
Thank you very much, Xavier. And there's a few minutes for questions from here in the room or in the audience.
Okay. Thank you for this nice presentation.
Thank you.
The mycorrhizal strain that you have been using is kind of commercial preparation, or?
Yes, this is a strain that was collected in IRTA and transferred to a company which is commercializing it.
Okay.
So this was originally collected under fruit trees, but since these fungi are not specific, they can be effective to inoculate other species. But it's a commercial species.
What about using isolating new mycorrhizal strains from dry area, for instance, to cope maybe a little bit better with extract? Do you have information in the literature about selecting mycorrhizal fungi from different area and testing them in?
So you mean selecting different strains which may
be more tolerant to drought?
Yes, we have some projects, not me, but with our colleagues, with tolerance to salinity, for instance.
Okay.
So the strains collected in plants next to the coast, submitted to high salinity, seem to have an effect of protecting the plants to excess of salt. We also have tried protection against heavy metals, and I think in drought also, in drought areas, which are more important factors to prevent drought in plants.
But do you have a collection of where you could try this?
Yes, some of them fungi are in collection, not commercialized, but we have the collection in careers. And to preserve this collection, what we do is to, because this fungi are biogrowth, obligate biogrowth, so we have to have the plant alive. But when the root is colonized, we can cut the plant, we can dry the substrate, and then this amount of root with colonized roots, soil propagules, and substrate can be stored for a long time, dried at 4 degrees. So we can have this inoculum for other projects.
And these other strains, they are commercialized or not?
Not for us, not for us. But well, there are several companies which commercialize different types of inoculum or mixtures of inoculum. For instance, then you can find in the market products with arbuscular mycorrhizal fungi and other biostimulants, but they don't specify which strains of fungi are using. Or even with the ectomycorrhizal fungi, which are much more specific and are very difficult to make it compatible. But well,
because with the change in climate, I think choosing the more tolerant strains which have been isolated from dry areas would be something interesting.
Yeah, in our projects, what we try to do is to use the commercialized strain as a control, and then we isolate strains from the area in which we want to work.
Thanks.
Any other question?
Hi, thank you very much for the presentation. It's very interesting. And I would ask, yes, did you apply the inoculation by spores, no?
Well, this is a mixture, the inoculation, and the inoculum is a mixture of, first, to produce the inoculum, we have usually these leaf plants, okay? And we inoculate them. And then after several months, when the roots are well developed, we cut the plants, we dry the substrate, and we mix all the roots, the substrate, and then what we are adding as inoculum is a mixture of spores, resistant spores, because when we dry, this fungi produce resistant spores, but also colonized roots, which also form resistance forms. And then we don't know exactly what we are inoculating here. This is a mixture. Since it's not a pure culture, we don't know exactly the amount of inoculum we produce. This is why we can quantify this inoculum and have some idea about the inoculation dosage. If we, with molecular methods, are able to calculate the amount of the mycorrhizal fungi, we use 2 primers, which amplify almost all the arbuscular mycorrhizal fungi, and then we can calculate with real-time PCR the initial amount of inoculum.
Okay, thank you. If there is no more question, we will move to the next presenter. Thank you, Xavier. She is.
No, you can present, yeah.
She is Bianca Maria.
Bianca Maria.
You know there was a Bianca there? Bianca Maria Senisa from the Catholic University. And she will present us the metabolomics in plant microorganisms interaction. You want to move there? You want to use your own computer?
Okay, so good morning everyone. I'm Bianca Maria from Catholic University of Sacred Heart, and today I want to present the role.
You have to share maybe the screen for the people that are online.
Oh, yeah.
To
Not connected.
You are not so
in the meeting.
Oh, you can connect or share with our team. You can share.
So good morning again. I'm Bianca Maria from Catholic University of Sacred Heart, and today I want to present the role of metabolomics in the plant microorganism interaction. So this presentation is mainly divided into parts. So in the first part, I start with a general introduction about the interaction between the secondary metabolites and the microorganism, and then I I will focus on some results obtained from metabolomics workflow and the role of metabolomics in the investigation of this interaction between the secondary metabolites and microorganisms. So the plants produce a huge number of different metabolites, mainly divided into primary and secondary metabolites. So while the primary are involved in the growth, development, and physiological processes of the plant, the secondary metabolites are not responsible for the growth and development of the plant, but are produced mainly in particular condition, generally depending on the environmental conditions. The secondary metabolites are small organic molecules with molecular masses lower than 3,000 kilodaltons and are mainly grouped into 4 classes, including phenolics, terpenes, nitrogen, and sulfur-containing compounds. The secondary metabolites play a variety of functions, are involved in the defense response signaling, in the response to environmental stresses, are able to repel the pests and pathogens, and act as signal in the symbiosis with the plant, between the plant and the microbes, and are able to shape also the soil microorganism. This relationship between the plants, in this case, mainly I focus on the roots. So the secondary metabolites of the exuded from the roots and the microorganisms in the rhizosphere are not unidirectional, but the secondary metabolites could shape the soil microorganism, and conversely, the microorganism in the rhizosphere could change the production of these secondary metabolites. So, in fact, several work reported that the microorganisms have a beneficial effect on the plant, may increase the nutrient acquisition, the response to environmental stress, modify the interaction with the mycorrhiza, and so on. But in the rhizosphere, the plants not only exude secondary metabolites and so phytochemicals, but also phytohormones that are able to change the soil, the rhizosphere microorganism community, including auxins, salicylic acid, and cyproteins. Of course, evaluating these interactions is quite challenging because, of course, there are a lot of other factors that impact this interaction. These factors include the soil type, so the chemistry of the soil, the plant compartment, so the leaves or the roots, of course, it depends. The host species and genotype, the plant immune system, and the developmental stage of the plant. And of course, not always these interactions are positive, but this interaction could be also neutral, which means that are neither beneficial nor harmful for the plants. But also, the microorganism could negatively affect the plant's physiological process, biochemical pathway, and the growth, and so be pathogenic. At this point comes into play the metabolomics. The metabolomics is defined as the comprehensive and quantitative analysis of all the metabolites in a living system, and the response of these metabolites to the changes in the environment. In fact, the metabolites are the downstream product of the interaction between the genes, the transcripts, and the proteins. And the metabolome represents the link, the bridge between the genotype and the phenotype. The workflow for metabolomics depends, of course, on the molecules under investigations. So starting from the sample preparation to the separation, the identification, and the data analysis, the workflow could be different. In our lab, we generally prepare the samples with methanol water solution, in an extraction with the methanol water solution, and then once the samples are prepared and injected in the instrument, we usually use a UHPLC, TOF mass spectrometer. The first step is the data processing. That is a fundamental step because here we obtain the meaningful information from the massive raw data acquired. During this step, it occurs the noise filtering and the baseline correction, the peak detection, the alignment of the peak and normalization. Then, of course, to reduce the amount of the data and facilitate their interpretation, we always perform a multivariate statistical analysis. In particular, we usually perform the hierarchical cluster analysis, then I'll show you some results, the principal component analysis, and these are both unsupervised analysis, and then the PLSDA or OPLSDA that are supervised analysis. So, how the secondary metabolism is affected by the microorganism. So here I reported some examples from our previous work, which underline the effect of sun microbes on the plant secondary metabolites. So here, as you can see, on the right, we have the unsupervised analysis, the HCR. So it's, and here is reported a heat map, it's called heat map. And as you can see on top, we have two different clusters. On the left part, the brown is the trichoderma, while in the second cluster we have the control, the Rhizoglomus irregulare and the Trichoderma atrovide. But this cluster is mainly constructed grouping the samples according to their similarities or differences. So as we can see here, the more similarities was found between the control and Trichoderma atrovide, and then with the Rhizoglomus irregulare. The treatment with Trichoderma atrovide, specifically on the seed, so the seed were mixed with the Trichoderma and then put in the soil, were the most different comparing to the others. So, which means that the secondary metabolism and the phytochemical profile of these samples were the most different compared to the others. On the right, we have the orthogonal partial least square discriminant analysis. This is a supervised analysis, which means that you insert an input to the software, but also the output to train the model to predict new data. So this plot confirms the results obtained by the unsupervised analysis. All these models are cross-validated, inspected for outliers, and the permutation analysis also is performed. And here we have the two score parameters, the R square and the Q square, that indicates the goodness of the model. So here the parameters are high because it ranges from 0 to 1. So are good parameters, so the model is good. Here's another example where we compare the Trichoderma with tomato treated with trichoderma and mycorrhiza, and only trichoderma and mycorrhiza. And as you can see here again, we have two clusters, so different phytochemical profile of all the treatment. Then confirmed by the unsupervised analysis. From the supervised analysis, which means the OPLSDA, we can also obtain a list of markers that are called BIP markers, which means variable importance in production. These compounds are listed, and here you have a score, which means that higher score, it means higher discrimination potential between the treatment. Another results we can obtain from metabolomics data is the pathway analysis. So we combine the fold change with the ANOVA and to obtain this graph. As you can see here, we compare for the treated samples with the corresponding control. And here you can see that the treatment with both trichoderma and mycorrhiza induce a positive modulation of the secondary metabolites of the samples. In particular, in the figure B, we can see that there is again a positive modulation of nitrogen-containing compound, phenylpropanoids, and phytolexin, in particular when treated with trichoderma, because the modulation is higher compared to those samples treated with mycorrhiza. That is the bar in red. Here is is online. So if you click on the column, you can also add the list of all the metabolites and the value of which they change compared to the control. Because here we represent this the sum of all the log fold change belonging to this class. So of course, to be more perfect, you can go inside the bar and see the compounds, the modulation of each compound. Again, to facilitate the interpretation of the data, another analysis we perform is called the Chemical Similarity Enrichment Analysis, or CHEMRICH. Here is the same as before, but all the compounds are grouped into classes. So here you can see a lot of classes, and the color of the dot represents the red is an increase of this class of compounds always compared to the control, and the blue, not blue, but the blue, which means a decrease of this class of compounds compared to the control. The dimension of the dot is the number of compounds belonging to that class. Okay, so these results suggest that, of course, metabolomics could be a useful tools to discriminate different microbial or fungi population, and how these microorganisms could affect the metabolism of the plant. But if we want to assess simultaneously in one experimental both the changes in the plant's metabolism and in the rhizosphere microorganism, we could also apply a multi-omics approach. This multi-omics approach is an integrated analysis that combines the data coming from different omics layers. So, through this approach, we can obtain meaningful information on the interaction between the plant, the host, and its associated microbiota. So in our case, we can combine the metabolomics with the metagenomics, but of course, in general, we can combine also with the proteomics data, lipidomics data, transcriptomics data, or also same omics coming from different analytical techniques. For example, metabolomics with liquid chromatography and mass spectrometry, and we combine it with data coming from nuclear magnetic resonance, for example. Okay, so what are the results we can and the information we can obtain from this integrated analysis? The first one is this arrow plot. Here is an example. The samples were treated with in stress conditions, both in drought and the combination of drought and heat. And as you can see here, in all the three components between the graph on the left and on the right, the agreement between the two data sets are high, because the length of the arrow is short. So the two data sets are highly correlated in all the three components, and so the agreement between the two omics techniques is high for both the treatment and all the samples. Another result is this circus plot. Here we're combining three different data sets coming from metagenomics, metabolomics, and the root exudate. So what these circus plots represent is the correlation between these two data sets. In red are represented the positive correlation between the features belonging to the different data sets, where in blue are the negative correlation between the features in the three data sets. Of course, here is only the representation, but we have which features correlated with which features. Okay. So to obtain more information on the variable belonging to the same biological pathway, another analysis could be the network analysis. Again, here we have in blue the data from the metabolomics analysis, the green is from metagenomics, and the red are the root exudates. And as you can see here, all the data sets are highly correlated. In red again are the positive correlated, and in blue the negative correlated. And the size node represents the degree of their interaction. Bigger interaction, smaller, lower interactions. So also here we have an example with the combination of metabolomics and transcriptomics data. On top, the samples were in no-stress condition when the figure C and D were in salinity stress. The sample were in salinity stress, and were treated with different biostimulants. One biostimulant and its fractions. So again, here we can see that the correlation between the two different data sets are high, because if you see the input in the block, on the left the metabolites and the gene, and the right the genes, the output is the same, both in non-stress and the stress condition. And this is confirmed by the cluster analysis on the right, the figure B and D, where the two biostimulants were grouped together and separated from the control, which means that both these biostimulants had an effect on both the metabolites and the genes compared to the control. From that analysis, we can also have more information about the significant feature, or in this case, metabolites and genes that were mainly responsible for the discrimination between the treatments. So, as you can see in blue, we have the metabolites and genes belonging to the control, while in the orange and gray, those belonging to the true treatment with biostimulants. So in the first case, in non-stress condition, the mainly responsible for the discrimination, the features were belonging to the control samples, while in a stress condition on the right, the bar are mainly orange, so the most discriminant feature were those belonging to the metabolites and genes in after the biostimulant, the fraction of the biostimulants. So here we have more information about this analysis. So to conclude, the combination between different omics technique could give us a more holistic view of the physiological processes involved in organism, and so we can obtain more meaningful information and the markers that are linked in different omics layers. Thank you for your attention.
So we have short time for some questions. If you, any of you have some, you cannot see if there are any. Georgios, I think, has raised the hand. Georgios?
Hello, good afternoon. Can you hear me?
Yeah.
Okay, thank you very much for this presentation. I mean, you know, it was exceptional. It was, from my opinion, more than I expected. Possibly some further observations our colleagues from ELGO, Dimitra, may be expressed. However, not getting into detail, as possibly Eduardo has notified you that we are planning a specific work, mainly for deliverable 5.3 that has to connect these physical attributes that you really were really interesting and that you presented regarding the network analysis, the correlations and all that, because these are methods that we are also using in the economic analysis as well. On the relation between metabolomics and economics, I mean, you know, specific properties of this. There is a correlation between these physical properties and their economic value. So this is a work that we would like to definitely proceed with. And I mean, you know, there are many things that I could ask and start a discussion currently on your presentation, but I would rather not do it now because I think that there's already a delay, and perhaps we can arrange a new specific meeting, because this is exactly what, in my opinion, exactly what we are seeking.
So, Georgios, have to be very careful about this, because there are really a few, if not correlation, between the metabolomics and the economic value at the moment. So what they see with this analysis, that plant or the microbe are changing their metabolome interaction that are involved in agricultural production. So, but to give a certain economical value to a certain metabolite, as far as I know, I was also talking with Michelle, it's not possible. I mean, maybe in a few cases, if they find, I don't know,
a certain bit, I mean, or whatever, but I would ask the criteria, that would be particular cases where you can find an economical value. Otherwise, it's just a change in the physiology of a plant that's happening, but there is no economical value. I just want to clarify this because otherwise we risk to getting into claims that we cannot sustain.
Well, there is a company in France which is extracting all kind of metabolites from many plants. And they use these metabolites for pharmacological questions. So it might be not related to our work, but in other situations, in other research domain, there can be some variation of these metabolites.
Yes, definitely. I mean, this on these observations and that Michelle is making, and of course Eduardo, because we have discussed it already quite a lot. And this is definitely an original research. I mean, it may not yield the results that we're expecting. That's an acceptable possibility. I mean, definitely. But what we're interested in finding out here, at least at the very basic level, I'm not saying that we're going to discover the whole chain of value chain, is that whether these microscopic interactions have a macroscopic effect, and eventually that can be further commercialized and have an economic value. This is a very basic research that is very important for us to do currently within SIRAM, because it will pave the way. I mean, it's not the end, of course. It will be a very basic.
Sorry, Jorgos, you need, to find at market level that somebody's preferring some kind of crops because it's got a change in the metabolism. This is not the case. That's the main point we got here. But yeah, there is also an attribution.
There's one short question.
Yeah.
Do you think it would be possible to select microorganisms, for instance, to increase in some particular kind of plants, like aromatic plants, to induce or increase the production of essential oils, for instance, which is a secondary metabolite? Maybe this could be interesting for the industry, or could it be possible to select, I don't know, fungi or bacteria which can induce this production?
Yes, we did a lot of experiment to try to see if different biostimulants in general could affect the production of these plant secondary metabolites, because we generally perform an untargeted metabolomics that is the full screen of all the metabolites in a samples. But we can also perform a target metabolomics, which means that I search for specific metabolites in the samples. We perform a lot of experiments, mainly, for example, on extra virgin olive oil, because in Italy we want to demonstrate that the oil coming from this specific region has higher value compared to those coming from other. So yes, it's we can also perform, of course, target, and I search for this. I want to and choose these metabolites as markers to also discriminate the best and the worst.
I can just comment very briefly then move forward. The part of your question related to the selection of microbes, this is what we do. In my view, it's quite difficult because when you select, I don't know, for nitrogen fixation, phosphorus, hydroxylic acid, you know better than me, it's quite easy to understand. Is this, for example, the case of induced systemic resistance? We don't know yet what is the determinant of the microbes. In many cases, induced, and probably the same is happening there, I guess. Yeah? So, and that is very interesting. There is a lot of line of research. Thank you. One.
It's a short question. It's about the detection limit in a complex matrix like salt, for example. In case you're applying this limit in soil, bulk soil example, versus the soil, which is the detection limit?
We have different extraction type depending on the samples. So in general, with roots, leaves, and so on, we perform extraction with the methanol and water solution. But for the rhizosphere and so the exudates, we only use water, and then we centrifuge, resuspend, and extract. So depending on the matrix, we have different preparation of the samples.
I was thinking on pesticides, for example, in case that the soil, we can improve in the relation of transformation products in pesticides in case of Regenerative Agriculture to demonstrate this approach. Depends on the detection limit.
Yes.
And extraction method.
But we work a lot with also pesticides, so we can also clean the. Yes.
There is a question from Lomna.
Yes. Yes. Thank you, Bianca, for this presentation. I have like a practical question. You said about multi-omic approach when you use it proteomic, transcriptomic, or metabolomic data. I want to know if we should have the same, do it, for example, transcriptomic or metabolomic or other omic techniques at the same time. We have the same sample analysis, or we can, for example, have an experiment, last experiment, have it the same condition, the same treatment, and we have it another result from metabolomic, and we can pull it the results or not.
Do you mean the data coming from the same experimental designer, or yes, you can combine also data from different, not the same? Because of course, the best option is to combine different omics data coming from the same experiment. Yes, because are more appropriate.
Okay.
Okay, thank you. We should move because we are a bit late. The next.
Excuse me, just a brief, Eduardo, because there was a brief interruption of the connection. Could you please repeat your last comment regarding my last question, if you?
Okay, yeah. So I don't want to be too much pessimistic, but of course it's better to start with the limitation. And the point was the fact that if the topic is to understand, is to gain an economical value due to the fact that you use a microbe in agriculture and the metabolome of the plant is changing, very easy. It's almost, I would not say impossible, but very difficult. We have seen other examples. For example, Michelle mentioned the identification of metabolites in plants, or maybe the fact that in fictional plants you get some interesting changes, and these are feasible. So I think we need to identify which are the aspects where we can do something meaningful and solid. Okay? I express in other words, but this is, I guess, the concept.
Okay, but I don't wanna. Okay, I don't want to insist on this one, but perhaps we should have a charting on this where we can on the limitations of the research, so that we can focus it correctly. But I would not insist now because the time is running late, so we would arrange a telco for this one. Thank you very much.
So thank you. The next speaker is Mark Viñas from IRTA. He will present himself better, and he will speak about the effect of cover crop fertilizer and different climatic factors on soil microbiota in vines. So when we are sharing, well, okay, when you wish, Mark.
Okay, can you hear properly? Can you hear me properly from the internet online?
Do you hear Mark?
No. So let's check one. Okay. Well, my name is Mark Piñas. I'm a researcher from IRTA, Sustainability and Biosystems program at IRTA. And we are going to present two projects, but they are fused or combined. One of them is a live project, and another is the continuation by means of the RegenCat project, the same project that Xavier told us before. The main aim here is to work on vineyards at field conditions, field tests, in two different sites in Catalonia, in Portla and in Espolla and Valles Oriental de Montbui. The two soils are different. One is acidic soil and the other soil is neutral soil. So the main aim is just to understand different regenerative management of the vineyards, mainly on the base focused on the green covers, but also in the tillage management and the fertilization. Sorry, because it's no, there's some problems. Okay, sorry. Okay, so the introduction. Well, it's all of us. I think that we are in a workshop regarding regenerative agriculture. So most of us know about the importance of microbial diversity. In fact, those diversity from the soil microbiota are really essential because the soil diversity they're really leading most of the soil functions that soils, as we know, is able to enable life on Earth. These ecosystem services, most of them are provided by soil microbiota. So the main aim in most of the projects in regenerative agriculture is to improve or increase this diversity and also their functions. Most of these functions are related with nutrient cycle from carbon, nitrogen, phosphorus, but also they are related with carbon sequestration, with microbial activity, but also the water and soil purification or to treat some pollutants in the soil. And in the end, also they are providing habitat for organisms. So other questions, important point is they're promoting soil aggregates and contribute to regulate the flood events, avoiding soil erosion and the loss of nutrients in soil. And in the end, all this genetic background we have in soil, most of these are related on soil microbiota. So it's really important to better understand the roles and how to improve or tailor them in the agriculture and regenerative agriculture. But the point here is that there are some factors that are really influencing or changing this diversity and functions. One of them is the intensive soil use, but also the global change, climate change, like rising of temperatures or changing the raining events and the erosion events. And obviously, the climatic conditions are very important and it depends on each site, so they have to understand and to be assessed concomitant to the microbial diversity. And in the end, crop and soil management are important because they are contributing at changing soil conditions and obviously changing this diversity sometimes at short or long term. Here I present some list of the main metabolic or ecological role that from soil microbiota it's the same, but they are all of them are related on ecosystem services, but mainly nitrification is an important step for soil health because they contribute to improve nitrogen utilization by crop production, plant production. So obviously nitrogen fixation, are also species here. They can contribute to the production of phytohormones, biocontrol agents. Well, I can skip because we're out of time, but this is an important point. All of them are related with microbial diversity and different kind of microorganisms. In fact, here it's just a list about how the complexity of diversity, because all of these functions really are related on different microorganisms at the same time. And the same function is provided by different microorganisms at the same time. Depending on the environmental conditions, we have one or other microorganism contributing to this function. So this is a complex because sometimes there's no change in diversity, but there are some changes on the function on the team. Regarding the state of the art of the advantage to use plant covers or green covers in for example, in binders, for example, or other crops, mainly there's different information, but most of them are confirming, that prevention of soil erosion is sometimes, depending on the soil, but may due to the depending on the conventional agriculture, when we don't have covers or we don't have some soil aggregates, but also at long term the vegetation water is important due to the carbon, organic carbon content, and also the soil aggregates, but also they prevent the dissemination of some weeds, for example. If we have a high diversity, we can prevent this, the occurrence of some weeds. But also they are also related, for example, in carbon metabolism, and as Xavier told us before, microbial necromass is one of the most important points, and currently it's an emergent topic because they have been described in different papers that about 50% of the total stable organic carbon, they are related to microbial necromass, and fungal necromass are quite important in the end in the stable carbon soil. We have to contribute to increase this fungal biomass as well. But here the point, the main objective in this work is to know more details on the impact of when we are using or we are starting with the green covers. The time, the age of these green covers can influence the soil diversity. It could be a time course transition, but also the fertilization of these green covers can influence as well the diversity as well. No? So this is not completely understood, and needs to be more research is needed. No? So these are the main objectives. To test different plant cover. But with different ages, but also combined with different fertilization strategies and different tillage strategies in two different soils, acidic and neutral soil. And also to identify which are the main phytochemical factors related on these changes of the microbial communities, and in the end to the functions that they can provide us by using different tools. Okay. Let's start with the first soil. I'm going to present the results of this soil mainly. This is from Girona in the north of Catalonia, and this is a Mediterranean region with low rain, annual rain, and with old vines, with 70 to 80 year vines. Goblet trained binders is a traditional binders in a soil, which is interesting here to highlight the pH because it's very low. From 5 to 6 is the pH of this soil. We have been tested 3 different conditions: the conventional management (CM) and the treatment by using green covers, young green covers from 1 to 5 years, and well-established green covers with more than 5 years. But we have been conducting 2 different sampling events of 2020 and 2023 in the framework of the 2 projects, MITMAC and AgriRegenCat projects. I'm going to present the first campaign because the second is already processing some results. We have been testing in the second campaign 2 different depths of soil, but now I'm going to present only with one depth, that's 0 to 20 centimeters. But we are interested to know the impact of this management in the depth of soil. Physicochemical soil monitoring is combined with microbial community assessment by different techniques and multivariate statistical analysis by analyzing the alpha beta. I will now show you the results. And the second side that we are starting now in the AgriRegenCat project is here in this site in Torre Marimón in Caldas de Montbui. In fact, the binders are over there, and they were conventional binders from 2009 to 27. But since 2017, no tillage was conducted, and since 2023, we started with other regenerative strategies. We have 2 plots at field conditions, and we have been testing different conditions, spontaneous covers versus sown cover, and also the different density of vines and different fertilization strategies, organic versus conventional, and also the impact of tillage, superficial versus conventional tillage. 4 sampling events are being conducted. One of them is pending, and 2 depths of soils has been studied with 6 replicates in each factor, condition, and time. And again, physicochemical soil monitoring by including micronutrients, micronutrients, carbon speciation, aggregates, and also this kind of carbon are also included, and combined with microbial community assessment by molecular methods, but also here function of soil, physiological function of soils by using ecoplates, enzymatic assessment of 30 different compounds have been assessed, and biodiversity of invertebrates are also being planned and conducted at different times in this project. Now I'm going on to show the results from the Girona site from the first campaign. We have all the process the results. What the main results regarding the physicochemical properties of soil after the 3 treatments, remember, conventional versus young and old green covers, we can see that the texture of the 3 in the parcels are quite similar. There are no significant statistical differences, but this is a sandy clay loam soil with more than 50% sand and more than 20% of clay. It's an important point because more clay, more microbial populations we have. Regarding cover crops, they showed an increase of some important nutrients, sodium, calcium, potassium, and copper. They could be related with the organic fertilization by compost. It could be related. And also we have seen important differences mainly in the young cover crop, with a lower plant density, where main could be that the compost was in surplus compost, and this excess of some organic materials could influence to some physical chemical parameters like the organic matter and organic carbon. You can see here. We have a very low range of carbon, below 1%, but it's higher in case of the young covers. So mainly by with this data, we can conclude here that the young covers could be more influenced by the fertilization. Both covers are fertilized with compost, and these dosage of compost have to take into account to improve this the impact of the can be in the soil. So it's important point to take into account in other experiments. Regarding the microbial communities in this experiment, again, we can see differences. In the young green covers, we have higher populations of both bacteria and fungi, and also with ammonium oxidizing bacteria, AOs, you can see. You can see that the bacteria in the MC, it's the young cover, it's higher in both in bacteria, fungi, and ammonium oxidizing bacteria, but not in ammonium oxidizing archaea. Yeah? Also, the cover crops, both young and old cover crops, impacts on the total bacterial population, increasing in a factor of 10 the total bacterial population in comparison with conventional methods, and reaching to it's not very high, but it's 10 to the 8, 10 to the 9, 16 copies by gram in the both treatments. Fungal population, this is an important point here. Here we have fungal. They are higher if we compare with the total bacteria, and in fact the fungi bacterial ratio are above 1. It's 25 to 22, and it's really interesting because in this soil, due to the acidic pH below 6, fungal population are really promoted. This is interesting because it's not common. So in binders, in this case, we have seen this. And in addition, it's also interesting that ammonium oxidizing archaea, not bacteria, are higher than bacteria. Sometimes clearly in this soil, and also could be due to the pH. You can see that we are to the 10 to the 6 in front of 10 to the 5 with bacteria. These are the main results in this case by qPCR methods when we assess the microbial population abundance. When we move to the metabarcoding results, we have been assessed both bacteria here by 16S metabarcoding and fungal population by ITS2 region metabarcoding as well in this plot. What we can see here just in the replicates is that when we are using, or we have cover crops, we have more homogeneous results in replicates than in the conventional methods. It's at phylum level, so it's an interesting point. But the bacteria, obviously, bacteria and cyanobacteria were the main phyla in this case, and there are some differences in the cover crops, in the old cover crops, promoting some groups, Actinobacteria, Cyanobacteria, and also in the young cover crops, the Comicobia bacteriota were also promoted. And regarding fungal community, we have seen obviously the Ascomycota, Basidiomycota are the main phyla, but Fusarium and Penicillium were the most predominant assigned genera. But when we have been used some specific tool to distinguish fungal biomarkers in each treatment by Lefse. This is a linear discriminant analysis effect sites assessment, just to identify the biomarkers in each treatment. In case of fungal community, we have identified some targets that could be related to the different treatments. Interestingly, Trichoderma were more related with the conventional management than by using green cover crops. So that's an interesting point. And Trichoderma and Penicillium was in the young green cover. When we assess the impact of this, the different age of cover crops in the alpha diversity, it's a way to quantify both the number of species of phylotypes, which is the richness, calculated with Chow, and we also quantify the diversity of ifness and richness by Shannon index. What we have seen is that again, cover crops promoted an increase of prokaryote in case richness, the number of phylotypes, but not the fungal richness, only the prokaryotes. So the prokaryotes are more more sensitive to the presence of green covers and also compost fertilization events. Also, only increasing in bacterial channel diversity, not weakness, was observed only when in the conventional methods. So, well, this is not true. It's the young covers. Right? And in the end, that no effect of cover crops were observed on fungal channel diversity. So we have important differences at alpha diversity, which is not very common in this kind of experiments, field test experiments. But here we have the hypothesis of covers. Moving to the beta diversity, which is it's a system where we can distinguish differences in the composition of the phylotypes we have in each sample, and we can compare this composition and the relative abundance of the composition between samples. We have to take into account we have more than 5 to 1,000 phylotypes in each sample, and then by beta diversity analysis we can compare all of them among them. We can see here we could see an important shift of the diversity in composition when we use the young green covers when we compare with both the other two treatments. So this was confirmed by Permanova statistical analysis to confirm that it was really an important shift in when we use this strategy. Again, we suspect of the impact of compost in case of young covers due to the lower plant density we have. And regarding the main environmental factors that they are contributing to the changes in the microbial diversity, this beta diversity we have seen before, pH, conductivity, and phosphorus content, but also organic matter in soil were really influencing in the prokaryote diversity. But interestingly, organic matter and organic carbon didn't impact on the beta diversity of fungal community. Fungal community was more related. Bacteria were more affected by the organic carbon differences we have seen in the physicochemical analysis. Now the last results we have seen, we conducted were first one to identify biomarkers, and the second one to identify the main biochemical pathways or ecological roles that could be impacted with the three treatments. Regarding the biomarkers, left analysis were carried out. This is a method described by Segat et al. 13 years ago, but some microbial biomarkers could be defined. Acidobacteria, when we use green covers, an important group, so group 10 was really a biomarker for young covers, but in conventional methods, without green covers, subgroup 13 of acidobacteria was also a biomarker. So we changed the biomarkers in the different treatments. But interestingly, it was a spirota which is related with ammonium oxidizers here, where a biomarker when the well-established green covers were settled out in the site. So green covers are improving this stable. These properties are really interesting because they improve the soil health regarding the nitrogen cycle. And just to conclude, the last method was FAPROTAX. This is a functional annotation of prokaryote taxa. In this method, which was defined, it's recently 8 years ago, more or less, by Luca lab, we can simplify the complex microbial diversity in their functions. So we can compare the functions in case of taxa that are cited at genus level in 80 different metabolic or ecological functions that are important in soils. The results we obtained with these three managements you can see here, and here we have seen just as a summary. We have there are a total of 80 functions. Here, I think there are the first 20, 25. But we can see that, for example, for fermentation, the green bar are really higher when we are using young covers, and with well-established covers and conventional methods, we have a lower fermentation capability. But again, with aerobic chemioheterotrophy or chemioheterotrophy, which obviously is the most common metabolism, are promoted or higher in the system, where we have young covers and the compost fertilization. But if we see in different scales, different metabolic pathways, we can see nitrification with when we are using green covers, and in the young green cover, we have been promoting the capability of aerobic and anaerobic mineralization and fermentation of organic carbon, but also aromatic compound degradation, which is really interesting to humic substances and complex organic matter for soil health. And a complete identification pathway has been promoted, metanotrophy, and also ureolysis, which again is an important step for soil quality. So by combining left and upper tags, we did a good strategy to simplify the complexity of the microbial diversity, and we can get some important ecological roles that could be or not promoted statistically way, not only just to this description, but also to demonstrate a potential contribution in this important biochemical pathways in soil. So as a conclusions, again, the climatic factors are important. They influence on the soil microbiota. We have a positive impact on soil organic carbon, micronutrients, microbial diversity, and also on potential soil functionality, which has been studied with young green covers, and could do by the compost addition, as we have told before, and again, cover crops and compost in acidic soil in Girona has increases the prokaryotic richness, the defined potential, but not the fungal diversity. And just to finalize that the doses of compost is important to be considered when we are starting or fertilizing green covers, and the further research has been conducted thoroughly at different soil depths and at different time course experiments to confirm these tendencies in these projects. And thank you for your attention.
Thank you, Mark. We are a bit late, but we have still time for a couple of questions if they arise here or.
Observation, very nice work. It's a rather technical question. I'm not familiar with these crop products while we use, as you probably know, Picrust. Yeah. Can you tell me what could be the difference between the two? Yeah, so if you tested both, they're very similar. Really similar, but crop products is better for ecological role. It's more. So there is a better curation of the function related to the ecology. Yeah, it's very interesting. This is being used by some soil labs. Probably. Yeah, Picrust is more on human microbiome. So yeah. Okay. Thanks.
And one question related to your question. Have you done a kind of similar work with fungi? Because there, probably there are some databases that are available with fungi or fungal traits that are allowing to assign functional traits to fungal. Gives some very interesting information about because your results are very interesting about the fungi. So with the difference in different genera that you can find under the different treatments, very interesting. But it would be interesting to know whether there are changes in the different functional groups of fungi.
That's you're right, but currently the available database is for prokaryotes, only for prokaryotes. It's other ducks. They are not related to fungal.
Yeah, but there are some fungal, some that
Yeah. That's a good point. You can try that. Thank you.
Any other questions? It doesn't seem that there is any from online participants. So, as it is late, I think we can conclude or close the workshop. And I want to thank very much all the participants and all the speakers and all the collaborators also. And hopefully we will get the presentations from everybody and we will share them with the people that has registered in this workshop. And that's all. I don't know if you, Eduardo, want something?
No. I just like to really thank and congratulate for the workshop because I think it's been really interesting and insightful. So I think we're doing research that is very interesting to see the correlation, the interaction with other similar processes. It's very good.
So that's it. Thank you very much, and hope it has been interesting. And see you very soon. Thank you very much.